Hi, welcome to Neuroverse, a podcast hosted by Carolina
and Clara, where we discuss all matters from neuroscience to philosophy and beyond. Welcome back to another episode of Neuroverse. Today, we're joined by Mateusz Kostecki, and we're going to be discussing neuroscience grassroots initiatives and promoting neuroscience tools and ideas. Mateusz is a neuroscientist and currently a postdoctoral researcher at the University of Heidelberg, where he studies early evolution of nervous systems and patterns of behavior in placozoa. Previously, he completed a PhD on social transmission of information in rodents at the Nensky Institute in Warsaw. He is also the founder and head of the Nensky Open Lab project, which is an international network of researchers and initiatives that promote the exchange of neuroscience ideas and tools through workshops and symposia. Today, we will be talking to Mateusz about Nensky Open Lab project. But first, let's start as we normally do by asking, how did you get into neuroscience in the first place, and specifically the research that you've done so far?
Hello, thanks for the invitation. I'm super happy to be here. Whenever I'm answering this question, how did you get into neuroscience, I always feel that it's always those stories, always some form of self-invention or a myth. So depending on the mood, I probably have different stories. But I generally started studying psychology and sociology after my high school. And I was kind of always interested in this old problem of nature versus nurture. So I thought that maybe I can find some answers for those questions in psychology, sociology. And then during my first year of my studies, I realized that I'm actually interested in biology, and maybe biology would provide me with better answers. So I switched my studies. I started studying biology, but I think more or less I always had some kind of random assemble of interest. I was interested in developmental biology at the beginning, in insects development, stuff like this. And I ended up doing neuroscience, I think, quite randomly at the end, because my colleagues organized some kind of like a neuroscience club at the University of Warsaw. It was like a student organization for promotion of neuroscience. They were organizing meetings every week. They were organizing symposia and conference. So they just kind of, invited me to come, and I came and I got interested. And that's more or less how my adventure started. It was a bit random, I would say. And then I was interested in multiple things, birdsong, courtship of Drosophila. I did some internships during my studies. But I ended up working on social behavior at Nensky Institute in the laboratory of Ewelina Knapska, where I think I decided to work on that project because I had a lot of freedom in this lab, and I decided to try to see maybe if I can observe behaviors that no one actually observed before. So I was studying how mice transfer information about localization of food in space. So it was a kind of, like exploratory project. And we found that actually, yes, they can do that, a bit like bees, but without the dense part. We still don't know probably how do they exactly do that. But yes, if one mouse finds food somewhere and it interacts with another guy, this other guy can kind of extract information about where this food was found by his colleague. And we did some calcium imaging on that. I was studying how the spatial representation is shaped by this acquiring of this information. But during my PhD, I kind of shifted my interest once again, and I was getting more and more interested in how it all started. So basically how the nervous system evolved. Yeah, it sounds a bit random when I talk about it, like it's completely unconnected parts, which I think the human biography is always at the end. So after my PhD, I just moved here to Heidelberg recently, half a year ago, and I'm working on those brainless creatures. I was actually supposed to do learning on them. I will probably come back to that, but my attention was just grasped by the weird behavior that they exhibit when you add some peptides, because they are using peptides to coordinate their behavior. So now I'm just like following my curiosity. I feel sometimes here like I'm doing 19th century kind of science. With just observing some random things and following them up without much consideration. Yes.
Yeah, it sounds really cool. And I don't know, it sounds like it is connected, actually. I think the theme for me was kind of learning and plasticity, but I like the myriad of interests.
Yeah,
Yes, it can be.
I think I've gotten already a little bit of an insight in terms of like your approach, in terms of how you like to do things with the way you described it is doing like 19th century science where you do just some observations and then you follow them up. I think that there's a lot of value in doing science that way, in a way, I guess, like slower paced rather than like trying to do a million things at once, throwing everything to the wall and seeing what sticks, and more of like a methodical approach of like, okay, we've observed this behavior, let's follow that, rather than being, I guess, like overly worried about what the path it's taking you down, like if it's like answering whatever ground you've applied for, whatever storyline the paper is about.
I think like my science is literally slow because my anima's extremely slow. Like I can record them with like 1 frame per second frame rate, so you can imagine how slow it is. Yes, but I think this is my approach to kind of let nature kind of lead you and not come to it with some like preconceptions. Of course, we always have some preconceptions. It's impossible to not have preconceptions, but kind of focus more on observation and letting the things surprise you somehow. I know it's not always achievable. I think I'm just lucky with my labs I'm working in that kind of encourage this type of approach. It's not always like this, I guess.
Yeah, and I guess it's not that common either for people to work on such different types of organisms. And so I think that the sort of exploratory approach and more conceptual driven research kind of shines through. And yeah, I wanted to ask then, how did you start Nensky Open Lab and what philosophies it sort of grounded on?
Yeah, so this is the project that I started, I think, in 2018. It's again like with this biography question, it's hard to talk about your motivations from the past because there are multiple, I guess, and it's again about selecting the one story that you that you want to tell. But I think, like first of all, we are always organizing something with this neuroscientific association at the University of Warsaw. But then when I started my PhD, I kind of felt some forms of dissatisfaction. So I felt that we lacked some kind of community at the institute, especially for our PhD students. This is the first thing. The second thing is that like quickly started to learn some techniques and open science tools that I used for my behavioral studies, like Arduino programming or Bonsai or whatever. And I felt that it may be useful for others. I liked teaching, so I thought, okay, maybe I will just start running some small workshops at Nensky and also maybe find other people that would like to teach and learn new things. So basically, like more like an exchange of skills kind of thing. But then it escalated quite quickly. So we started to organize summer schools when the goal was basically to teach people more or less everything they need to learn, basically build their own behavioral setups and analyze the data. So we taught them how to program Arduino, use electronics, 3D printing, and then how to analyze data with things like DeepLabCat. So everything was kind of focused on open science because I'm not like obsessed with open science. I think it can become a goal in itself sometimes, but I think it's basically methods that are free or very cheap. Let people go outside of the constraints that are usually imposed by the commercial equipment. So they give you freedom. I think that's the important part about open tools. But what is, I think, very important is that tools are just something that allow you to answer your questions. And the more tools you know, the more free you are in the way you approach your question. So basically, if you are just limited by commercial equipment, you're also limited in the number of questions you can ask. So this was the main idea, I think, to give people tools that they can just use to answer their more crazy questions. Because if you can build a setup by yourself, you are not really restrained and constrained by what your lab has at this moment. And we started collaboration with people from Argentina, and now we are running our like biggest event probably is this transatlantic behavioral neuroscience school. It was called Summer School, but we are organizing it interchangeably between Poland and Argentina in Polish summer, which is winter in Argentina. So we dropped this summer part. So yeah, now we are organizing this big course when we really try to give people really everything they need to build their own setup and then analyze the data in more complex ways than what is usually done maybe.
So I just wanted to say, well, firstly, really cool. I really like the idea, especially in terms of the mindset of if you have the tools, then you have the freedom to do it. Having joined a lab that was brand new, I've had to help build everything from scratch, and I do think that gives you a lot of freedom in terms of being able to tweak things in your own way.
Mhm.
How is your network different from existing networks? Like, what makes it stand out? Is this idea of focusing on the tools as a method to provide freedom, or is there more aspects to it?
I've never really thought about what is different. We just run this, and many people who attended our workshops told us that they would like to run something like that in their institutions. So I think it's more like natural growth. So we never kind of compare ourselves to other projects. But I think what is kind of unique, if I would have to answer this question, is basically that we are not only focusing on these tool parts, which is like probably most important part of what we do, but we have another branch. At some point, we felt that, yes, exactly as I said, kind of tools are nice, but without proper ideas for what you are going to do, they're useless. And then that's why in 2022, we started something that we call School of Ideas in Neuroscience, which is like a very small workshop. So it's like for 15 participants, but we invite philosophers, neuroscientists, anthropologists, and the idea is basically to discuss, first of all, discuss different theoretical frameworks in neuroscience. So let people orient themselves better in this like landscape of theories that they may use to put their research in kind of more like general and more meaningful framework, but also to discuss neuroscience from a meta perspective. So, for example, why we are doing what we are doing, how discoveries are made, how ideas are born in neuroscience, and how people can learn from others to basically improve the way they frame their questions. For example, how to sharpen your questions and how to design your experiments in a better way. That was the idea. So we are organizing this school every year now, and then we also started to organize workshops of ideas that are more focused on specular ideas. So, for example, naturalistic behavior. In Italy in 2024, we discussed the concept of naturalistic behavior. To what extent should we care about if our behavior is naturalistic or not? This year we discussed what does it mean that the brain encodes and decodes information. So we are also trying to kind of, take a look at the popular topics in neuroscience and see to what extent they make sense, which is, I think, the thing that is kind of peculiar to our project. People do that, of course. I think your podcast is one of those projects that actually focuses on talking about neuroscience in like more general terms and from a meta perspective, which is amazing. And yes, we are also kind of in this vibe, I would say.
Yeah, I really like it. And I also want to say a couple things. Firstly, going back to the tools and the importance of making people realize how things are built from scratch and, the variety of tools that people can work with, not just like the ones that are given or the ones that are trendy. I think it's so important. And I also had a taste of it at a summer school, which is actually where I met Mateusz, where he was a TA and introduced me to bonsai. And I do no behavioral research. So that was really, really cool. And honestly, changed my whole perspective. So this is just my personal experience of being on the other side of it. Because, yeah, I felt really empowered by the whole experience because I'd always been so intimidated by behavioral research because I thought that it was really complicated and you had to have these like complex rigs and everything had to be pre-set up and softwares. And then I got a taste of it in the summer school and realized like it doesn't need to be complicated. And actually, when you break it down, it's pretty simple and anyone can do it. And so I think on top of the actual work that can be done with these tools, it's also the mindset shift in people that really makes a difference into what sort of questions they then go on to pursue to ask. Because now, for example, I'm way less intimidated by behavioral neuroscience. So now I see even more how important this is. And yeah, I love the School of Ideas concept too. And I also remember that like one of our first conversations was me realizing what strong opinions you have about play cells. And and then you mentioned how you had a symposium on some topic related to play cells or encoding in the brain. I think it was that one.
Yeah, this is the decoding and encoding case.
Yeah, yeah.
Yeah, we kind of try to be provocative in the topics of workshops that we organize. So when we organize a naturalistic behavior workshop, this is something I think super popular nowadays to encourage neuroscientists to make their setups more naturalistic, to focus on ethological relevance of their behavioral experiments. And people would say very often that like having a head fixed mouse and just showing like simple visual stimuli to it is not really naturalistic and will not lead to any interesting discoveries. And we kind of decided let's have more like a critical look on this fashionable approach. And we really had it. I think we wrote a preprint actually from that workshop when we our point at the end was that like what is important is your research questions, and you can approach it from very different angles, and from completely unnaturalistic experiments you can actually learn many things. So you shouldn't be intimidated by people telling you that your setup is not naturalistic enough. uh, yeah, so we try to kind of look more critically on things that are like widely accepted, which is I think always a good approach. So even if we are wrong, we at least let people to kind of look at things from different perspectives.
Mm. Yeah, and how did you actually go about starting this initiative?
Uh, yeah, I think like it was easy to me to go from those like student activities just to that. So now what we are trying to do now is basically to create this network of open labs. We have one at Nensky, one in Cambridge, one in Heidelberg now, and one in Buenos Aires. And now I'm writing some kind of like a brochure explaining people how they can organize something like that. And I think the most important thing if you're starting something is basically to start with whatever you have at hand. So I just had some Arduinos in the lab and I knew how to program them. So I decided I think I will start this initiative and I will start with Arduino workshops because I can teach people how to do that. I have some equipment. So let's see what will happen from that. So I had this like very basic thing. So I had some resources and I just try to apply what I have already. And then I asked my institute to give me some money and they agreed and then it expanded. So basically we started with extremely basic things and then we slowly expanded. So we got some money to invite people from other countries as well. And we got more international with time. But the starting was just basically, I have this, what I can do with this. I can do some simple workshops on Arduino, let's do this. And then when you already run some workshops, you can actually show it to someone and say, okay, we run those workshops, maybe we can get some money to do something else. So it was more like step-by-step approach without really much of planning and thinking where we can get. So I didn't plan to make it like a large international organization with 30 people. It was never a plan. It just happened, I would say.
And how do you find it changing over time? Do you think that because it's like tools focused, it's quite dynamic? Yeah, like with modern tools becoming available and things like that, or is it instead kept the same because it's like breaking it down to the basics that remain the same? And also like with the discussions, do the topics change a lot over time because of what's new and what's the most important questions?
I think for the Schools of Ideas, we are running it since 2022. And I haven't thought about like to what extent our topics of our discussion changed. I think at the beginning, maybe we were more eager to discuss things like predictive coding framework. Now it's maybe less of a thing, but I'm not sure if it reflects some real change in neuroscience or it's just our personal thing. We are mostly focused on behavior at the beginning. Now we are, I think, getting more and more into the brain. And in terms of tools, so we are teaching people how to use Arduinos, DeepLabCat and stuff. So things like DeepLabCat, of course, they change because if you don't know, it's a software that you can use to track body parts of an animal. So it's getting improved and improved over time. And we are trying to teach people some new methods, but it doesn't change so quickly. On the other hand, things like Arduino, they don't really change that much. The applications change. So we can basically show people how to control some new type of device with Arduino. Bonsai, which is a software that you can use to like design your behavioral experiments, run them, and then analyze data. It also changes, but it changes slowly. So the things that we teach are not that quick to change. We run some microscopy workshops, for example, every year, but there we basically teach people basics of optics. So like what are photons, how to build a fluorescent microscope. So those things also don't change. But yes, some things don't change that much, I would say.
So going back to the School of Ideas aspect of the Nenki Open Lab. So in your website, you say that we are not taught how to develop ideas and integrate our research with the existing knowledge. And at the school that you run, you aim to teach participants how to think theoretically and collect data that makes sense to be collected, which I really agree with. And I was hoping to dive into a little bit of the particularities of this. And I was
Mhm.
hoping, perhaps based on your experience and some of the workshops that you've ran, if you could give us a few techniques or exercises that you could describe that perhaps our listeners could take home and practice and try and develop a little bit of that. In a way, like critical thinking, but not, as we mentioned, just about the tools and exactly like what we can collect with the tools, but what we can do with all that extra knowledge and ability.
Mhm. Oh, it's a hard question. So like we organize the school in the way that, first of all, what we do is that we don't give people like 15 minutes or 20 minutes for a talk, or not even an hour. We have 3 hours long talks, of course, with 2 breaks in the middle. So one thing that we do that's hard to replicate back at home is basically to engage with a very detailed discussion about for example, some research that someone is doing. So even if we have like a more research talks during the school, we really discuss things in depth. So like, why did you do this? How did you come up with this idea and stuff like that? So we kind of try to look really closely at some piece of research. This is also the way you really practice like developing ideas by yourself, but also like critically looking at research in general. So this is one thing that is important in those schools, that basically at length discussions are super important. And that's the thing that you can basically start maybe back at your institution. Now every 2 weeks we run something that we call School of Ideas Journal Club with our friends, when we talk about papers more in this way. Like we don't focus that much on the, like, for example, which virus was used, or like what light intensity, but like, what does this paper tell us about reality? Why was it done? For example, we'll be discussing the new International Brain Laboratory paper soon. So I'm super excited about this. So this is something that you can do, basically organize something that is based on discussing those whys of papers. During every school, we also run group tasks, but I'm not sure to what extent you are able to replicate that, but because what we are, for example, doing is that we are asking people questions about their intuition. So this year in Buenos Aires, we ran a workshop about variability of behavior. So we just ran a survey when we ask people about which behaviors do they think are more robust or less robust. So like basically how often you will see certain behavior in your experiment. And then we discuss those intuitions, because it turns out that people will have different intuitions, which is basically a task that shows you that people have some preconceptions that are very often not verbalized, and they influence our methodological choices. So for example, if you assume that feeding or sexual behaviors are absolutely not learned, you may approach them in a different way or be surprised by different things than if you have this feeling that maybe there is some kind of plasticity there, for example, or some learned component. So we actually like to probe people's intuitions. And this is something that you can try to also do by yourself, to observe sometimes why I thought about things this way, or how do I differ from my colleagues in approaching this research project, for example. It's very nice to see, for example, when I'm in the lab that is mostly doing evolutionary biology, I'm in fact the only like systems neuroscience in my lab. And it's very funny because we see different things. Like we look on the same data, and I see, for example, some oscillatory patterns in behavior, let's say, that my colleague also saw, but they were not interesting for him. They were interesting for me because I read about oscillations or whatever. Yes. So this is very funny how your background can influence your research. So I think those things can actually, you can try to look more closely about your preconceptions and your biases, which is actually kind of fun. During all the workshops actually that we organize, we realized that one thing that comes up every time we talk about things like naturalistic behavior or decoding and encoding information from brain activity is that people very loosely define the concepts they are using. So very often we are talking about naturalistic behavior, we can have completely different definitions of that. So for some people, naturalistic behavior may be just like spontaneous behavior, or like even freely moving mouse in an open field can be naturalistic for some people. And for some people, it basically means a mouse running in a forest with some kind of non-invasive recording device. So one thing that is always can be interesting for anyone is to basically try to think about the concepts you're using in your research. Things that may not seem too important, like exactly like naturalistic or decoding, or even spatial navigation, or like social behavior, and try to think like, how would you define it? So what do you mean by social behavior, for example? Would interactions between bacteria be, for example, for you a social behavior or not? Now I'm working with those guys that don't have brains, and they also have some form of interactions between each other. Do I call it social or not? Sometimes it doesn't matter, and I think it doesn't matter in most of the causes. I don't think that definitions are that important, but sometimes they really can generate some kind of misconceptions and conflicts that are not really there. And so actually, I think it's a good exercise to try to think about the definitions of concepts that you're using. Yes, and thing that actually I think I can always recommend is to read a lot. So it is always a thing that is useful, and read not only things from your field. First of all, read old papers, because we many times when we had some discussion, we realized that something was already really well discussed in a paper from 70s, because back then people had more space to write. Like when you are like doing experiments, you describe your experiments, but you had more space for discussion, so people were discussing things more in details. But also read things from, like, I think to some extent it's nice to get acquainted with some philosophy. For example, like even neurophilosophy, so philosophy that is directly connected to what you are doing in neuroscience, to papers that discuss, for example, causality in nervous system. What does it mean that something causes something, or mechanism? So what we talk about when you talk about mechanisms. So for example, if I'm studying mechanisms for spatial navigation, what do I mean by that? Yeah. Can I really do that? Is it achievable to study mechanisms and stuff like this? So that's what I would recommend, at least from time to time. And that's what I think is useful, because it kind of gives us tools to discuss our things more precisely with other people. And so reading is always recommended. I think it may seem trivial or pretentious, but it really works. And one thing that also came up multiple times during our discussions is that there is this large piece of knowledge that we generate in our labs. It's maybe less related to ideas. It's maybe to some extent related to those tools that we are using. There's a lot of knowledge that we do not really acknowledge in paper. For example, in my case, I know that my experiment works better when the light is off, and I will probably not write it in a paper. This is things that we skip. For some people, for example, experiment might work only on Tuesdays, or depending on how they are dressed to work, or if it's male or female experimenter. So there's a lot of this knowledge that we don't put into the paper. It's called implicit knowledge, that may be important to really replicate our work, or to understand our work properly. So it's very funny that, like, probably on all workshops, this topic came out, and people started making notes about those things, and making more precise descriptions of those things that we usually don't put in the paper. But this is something that really came up on last few workshops, how important this implicit knowledge. Some of them are just superstitions, but they may matter.
Yeah.
Yeah.
No, it's really interesting. And I like the point that you made about, like, we all have base assumptions that we assume to be true, and then we set up our experiments with that in mind. And it is really important to like stop and question those things. And I don't know if you feel this, but I also find it cool that you have this big international rapport, like across Argentina and different universities. And so I wonder if that also helps bring in, some perspectives outside of the box to be able to like challenge you a little bit. And if different perhaps cultures or labs have their own intuitions on how to do things.
Yeah, I'm so glad that you mentioned that. And yeah, this kind of idea of experimenter bias came up a few times in our discussion today, which is interesting. And like I just keep thinking about we have like coffee in our center, and the other day someone made the coffee and like wrote by the coffee. This worked even though the coffee machine seemed to be broken, question mark, question mark. And I just read this and I was like, this is hilarious because it looks like what I would write in my lab book. Like, I don't know why this person wrote down all the description of their coffee making process when the outcome was just fine and they managed to make the coffee. But I was like, this is my lab notebook.
Mm-hmm.
And it's true. I do it too. And I'm always like writing down every single thing just in case I need the information, just in
Mm-hmm.
case I need to compare why this worked and why it didn't. And I never use this information, but it is so valuable.
Yes. So I think that was exactly the thing that we discussed in Buenos Aires last time, that people make those notes sometimes, but they never really come back to them at the end.
Yeah.
So I was also making those notes, like this mouse seemed a bit, I don't know, tired or whatever. So maybe if I will be interpreting the results, I will take that in consideration. But I don't know, how do you feel about it? But then how do I quantify my impression that mouse was probably tired? Like, I cannot quantify it. It's probably not enough to exclude the mouse from the experiment. But like, does it happen to you actually at all to like really come back to those notes and use some kind of this contextual information to interpret the data, for example? I'm not sure.
I mean, I think sometimes it might, like, if there is a difference between like say two brains or two animals, then you might go back to your notes and realize like, yeah, it's happened to me before where I've gone back to my notes and been like, okay, potentially it could be because of this one thing that seemed to be happening always in this group and not in this group. So I think it's always important to just write it down just in case. But
Yes,
also I
yes,
think
yes.
it's just nice to highlight like the reality of biology and the variability in biology. And this is something that maybe people don't realize when they go into science as specifically become like biologists, because your experiments are not all in these precise confined environments in a vacuum or
Hmm.
whatever. Like it is subject to nature and variability, and that's just science and that's part of the fun. But it's also like, yeah, remembering to take things into account that you might not initially think maybe influencing your results or maybe the cause of whatever. Yeah.
Yeah, you're mostly doing physiology, am I right?
I do imaging and patching. Yeah.
Yes, because like in behavioral studies, the basic assumption is that like behavior is variable and context dependent. So it's like somehow, uh, this like kind of common knowledge that behavior is variable. And people who work in behavior for a long time are not surprised by variability. But I sometimes notice that people coming from like engineering field, the beginning they're like, what's happening? Why it's so variable? I was expecting more like robust results. And I'm not sure how is it in different subfields of neuroscience. So for example, if you're working with patch, so to what extent you are used to this context dependence and variability
I mean, so much. Like, I mean,
so much?
it's like I do patching in ex vivo slices, and like the temperature of the bath can influence the membrane dynamics and therefore the cell's
Hmm.
activity. So
Hmm.
it's like there are like 1,000 variables. Every single thing can influence the recording, and
Hmm.
the temperature that day can influence how the pipette is pulled, and how the pipette is pulled influences its shape and its resistance, and that influences
I
how
see.
your
Hmm.
patches, and so yeah, a lot.
I see. So like in your case, like a measurement method also introduces a lot of variability in itself.
Yeah, exactly. There's
Hmm.
like the variability of the system on its own. And in that also I experience a lot of variability because like I'm expressing things using viruses, which are inherently variable. I also find
Hmm.
like when I try to be more controlled with viral expression, it doesn't necessarily work better. Sometimes you just need to gauge concentrations based on the outcome, not based on like the specific titer, dilution,
Hmm.
whatever.
Hmm. Hmm.
Um, so yeah, there's the variability in that, and then there's the variability in the measurement system, and so all together,
Hmm.
it's
Hmm.
yeah.
Hmm. It's very funny because yeah, in your case, it's like also maybe it's easier to pinpoint the sources of variability. So you can say that like a temperature will influence some biophysical processes, but in behavior, very often you just don't know. People just like, "Ah, it's just mysterious. There's something happening in the animal." People like often anthropomorphize animals in this way, saying like, I don't know, they are tired or they're bored or whatever. But it's not really known what is causing this variability. And Carolina, your field is what?
Yeah, so I train mice to do a specific task, and I use two photon imaging and neuropixel
Hmm.
recording.
Hmm.
Um, and while I agree that yes, like behavior is extremely variable, I also think it can also be incredibly precise. Like if, for
Hmm.
example, you have a space that is off by 3 millimeters, the mice will no longer engage. So like it can be down to like a couple millimeters of the setup, and you think it's basically the same thing. Like, why are they not
Hmm.
engaging? But those like slight tweaks make all the difference. And I do think
Hmm.
it's really cool in like projects like the IBL or with really well planned and studied behaviors like the bandit task, that we can reproduce a lot of the behavioral data and results and paradigms. And you'd think that even though behavior is extremely difficult to get to work and it's extremely variable, it can also be reproducible. Yeah, that's also in a way the wonder of neuroscience, because I do look at the behavior and I also sometimes think like, how will this ever be reproducible?
Hmm.
Specifically with my PhD, because I'm starting a behavioral paradigm from scratch. And so it's just been a lot of iterations, whereas when it's already set up, it's already so stereotyped and it works so nicely and it's so
Yeah,
reproducible.
that's amazing. Like my behavior from my PhD was also super stereotyped. Like it really worked every time I tested that. So
Hmm.
I'm talking about variability, but in fact my behaviors were more or less robust. But it's very
Hmm.
interesting what you say about also IBL task, because I think it is a proof that actually if you spend a lot of time and you will have many people and a lot of money, you can actually standardized things. So many of
Yeah.
those things that we just assume they're like kind of magical, we don't know. This is like those capricious animals. Is basically this thing that you say it's like 3 millimeter space that matters. And if you don't care about it, you may actually see variability that is just related to that. So it's a very good point. I always wonder just with IBL task, to what extent we can really afford doing that every time we are doing behavior. Like it's a one huge project that took many years, a lot of money, and we cannot do it with every behavior we are studying. But I think it's a very nice example in a way, like that you can standardize at least some simple task, a relatively simple task. Of course, it's not a simple task. I really like IBL also as a thought experiment that it shows
Exactly.
that you can actually standardize things. And
Yeah.
sometimes this mythical variability of behavior is just, yeah, it stems from like very concrete things that we can pinpoint if you put enough work.
I do think that the IBL task is, on the one hand, like a very, very expensive thought experiment, but at the same time, very needed because before that, I think we never really knew how reproducible and standardized behavioral neuroscience could be. And I think now
Hmm.
we're a lot more confident. I guess, like, I don't think it's feasible to do this for every neuroscience lab or experiment or paradigm because, the main constraints with having something as standardized is you're so much more limited in the type of questions that you can ask. So I do think it's about being able to say like, okay, like neuroscientists can standardize behavior, and being able to show that you are a good enough experimentalist that you can almost like project a similar level of confidence based on your behavioral results and setup as the IBL has done with their own. So I think that's where behavioral neuroscience needs to get to for everyone to be able to like trust what the results that they're reading.
Yes. Yeah, it's also actually interesting when you compare it with this approach to like more naturalistic neuroscience, when you are basically losing control at all. Like so IBL is this like super control task when you basically control this spacing of 3 millimeters. And then if you are approaching neuroscience from this naturalistic perspective, you just say, okay, I'm putting mouse into the forest and I don't have control over anything. But then I assume that maybe the more like real patterns of activity will be revealed. So it's funny, like those two different approaches. Yes, I think like I don't want to say something banal like the truth lies in the middle. I think it just depends on your research question. Both things are actually valid depending on the question. But it's always to me interesting to talk with IBL people and then talk to my colleagues who are like studying animals in the wild, how different those environments are. Even the culture seems like slightly different between those environments, and I like both of them. So
Also, just to clarify, when you were speaking about stereotyped behaviors, are those trained behaviors like in a lab? Because obviously there are also stereotyped behaviors in the wild, but maybe they're just like less precise. Like maybe they're not always driven by a specific distance of less than 3 millimeters.
Yeah, sorry. I did mean more like reproducible and learned behaviors in a lab. I guess yes, stereotyped and reproducible behaviors also happen in more naturalistic setups. I'm just so geared to think about it in the this more like head fixed, less naturalistic way. I do think the grass is also always greener, on whichever side you're in. Like I look at naturalistic paradigms and I'm like, wow, wouldn't it be nice not to have to train my mice every day? But yeah.
yes. For the first time in my life, at least I'm working for like half a year now with my animals. I feel that the grass is super green where I am. But I know this like feeling of like, oh, maybe I should just, okay, go into the forest with my recording setups. But yeah, as you, Clara, say, of course, like, yeah, because you have this stereotypic behavior that you train, but of course you will have those stereotypic behaviors that are so-called innate behaviors. I don't believe that this word has much sense, but reflex-like behaviors would be also stereotypic, I guess. And you can find them in nature.
I wanted to ask, I feel like we've gone a bit off tangent, which I'm totally happy with, but I just wanted to ask, going back to the Nensky Open Lab, if
Mhm.
any of the workshops or symposium, which I'm sure by now you've been to so many and helped organize, but has there been any particular moments of realizations or discussions that have influenced your own research?
Oh, multiple. Even the fact that I'm here is caused by the discussions we had last year on the School of Ideas of Neuroscience, because my current PI, Gaspar Kelly, who is actually working on this early evolution of the nervous system topic, was a guest last year. And we had just like after the school, we had a party at the beach, and I discussed with him some things related, for example, to what he said during the talk about placozoan behavior. And then I really got hooked on an idea on studying those animals. So very directly it influenced like even the choice of my animal model. But yes, multiple times, like I had, of course, purely tool related epiphanies. So for example, realizing that I can use this concrete tool or I can build this device in this way. Yeah, it happened to me multiple times. Or I could, like, for example, once during the 3D printing workshop that my colleague was running, I just realized I can very easily design a mounting for my specimens that would basically speed up my work. So things as easy as that. But also during the School of Ideas, I got multiple ideas that then I used in my research. So even my project now, which is basically I'm studying also how temperature is influencing the speed of oscillations in behavior of my animals, is directly inspired by a talk by Eve Marder, who was a guest of our school in like 2022, and she was talking about how temperature influences the activity of neurons in stomatogastric ganglions of crustaceans and how robust those oscillations are to changes in temperature. And then when I started working with my animals, I had this like, oh yes, of course, I need to do that. Like when I saw those oscillations, it was my first thought. It was like deeply ingrained in my memory. So yes, in many, many ways, I would say those things influenced my research. But also like the people I met, I still collaborate with them on some smaller projects, or like we discuss a lot together. And it still kind of is influencing what I'm doing. We really would like to one day run a survey and ask our alumni of schools and workshops in what way it influenced their research. We have some collaborations that started between people. There's a paper that will be published soon that stems from the collaboration between my colleague Kacper and people from Cold Spring Harbor. Yeah, but that's really something that I would like really ask people and see those answers, to what extent also influenced other people, because for me it was super important.
Yeah, that's really good to hear. I mean, I expected that, but it's nice to hear how direct it was and that the concepts and ideas actually come to fruition in research.
Yes. I think like when I was younger and I was starting this initiative, I had this impression that ideas in neuroscience and those theoretical frameworks are super important and we should spend more time on focusing on theories and stuff. I think after all those like 3 editions of the school, now the 4th is running in Warsaw, I realized that I think they have more like a modulatory influence over what we are doing. So reading about more theoretical frameworks or like some philosophy or whatever will give you some ideas how to push your research in this or the other way. It's not directly really like changing the way you approach your science, but I think it's a reservoir of ideas, and yeah, it's more like modulatory influence.
Yeah, I like that.
Well, similar to this, but also related to what we were talking about earlier in terms of broadening your horizons and reading lots of papers, not just within your field, but also other fields. Do you have any advice in terms of, I guess, how to remain curious about the science that you're doing and other papers in general? Because something that I've noticed a lot with scientists, myself included, is you get so bogged down with your work. It's really hard sometimes to like look up and think about what you're doing, or to even be curious about not only your work but beyond. And I think most of the good ideas come from mixing fields and being exposed to different things. And I think that sometimes the work, it can kind of like dampen that curiosity spark, which we all have coming into science. Do you have any advice for being able to maintain that curiosity, to being able to keep testing your base assumptions, which I think is incredibly important to do all the time? Yeah.
I would say, like, I would start from the other side. The first thing that I would like to say is that I would really like to tell everyone that you shouldn't feel guilty if you don't feel curious anymore. And I know only very few people who remain interested in their project at the end of their PhD. I think like this is the moment when people usually really are like frustrated and tired and they stop reading more things. And it's absolutely normal. And I think it's normal to have those long periods, even when you are not interested in anything and you don't read much. And it happens a lot. It happens a lot to me. But I think what is useful, or what was useful for me, for example, I think Roman Brett in his interview you in Brain Inspire podcast said once that he has Wednesday is this day when he basically reads papers and thinks more about his research from this like theoretical perspective. I cannot afford spending whole Wednesday on reading. I would also not like to do that, I guess. But I now try to every like Wednesday morning for at least 2 hours to read papers that are like not exactly strictly related to what I'm doing. So this is the thing that I started doing, and it seems to work for me. But of course, for me, what worked was basically to change my field and like start doing new things. So then your curiosity is always fresh. But I think like finding this time that you spend on reading, and it's really time for that. So you don't do other things if you can, of course, afford that. The problem is that, of course, not everyone can afford if you are training your animals for 12 hours daily, which I hope I will never have to do. And I will do everything not to have to do this. But if you can just afford like to select some time in the week just to read around, this is one thing. But I also feel like this is a very social thing. Interest is very social for me, at least. And like having a group of people with whom I can discuss things. So we have those journal clubs every 2 weeks with my friends whom I met on the Schools of Ideas. We discuss a paper every 2 weeks. And it's also a time that is just spent on speculating and discussing things that are unrelated to my field. And it really keeps me going. So I think like finding a group of people who share your desire to stay curious is always useful. I think that's more or less the goal of Open Labs also, like creating Open Labs in different places so that people can like work together, like discuss things together. So like keep this flame going, let's say. But also, as I said at the beginning, if you don't feel curious anymore, it's fine. It happens. So many people who are like reading a lot and they were interested in multiple things that I knew, they stopped reading anything besides like papers related to their project at the end of their PhD, and they were super bored with them also. It's fine.
Yeah, and if you're really desperate, also you can always go to a summer school, as I did.
Yes, of
And it
course.
definitely will rejuvenate your curiosity if it's a good summer school.
That's the hope, yes. Actually, I think many summer schools, like even tools related, are helpful. You have this experience of being among people who are sharing your interests, and it's also, like a lively environment to discuss things. That's why I think like those things are important. But like if anyone would be interested in running things like we are running, you can just contact us because it's like very easy to do. As I said at the beginning, you can just start with whatever you have at hand and start organizing some workshops. We, of course, encourage you to check our social media or our webpage to see if there's any event that may interest you. Because like we try to really organize workshops that are like useful, and we try to keep them as cheap as possible or free even sometimes. So yeah, if you really need training in some specific thing, especially related to behavior neuroscience, you may find our events useful.
Yeah, we'll definitely include all of the details about Nansky Open Labs on our website and on our social media as well, and in the episode description. So it should be easy to find. Yeah, thank you so much for joining us today, Mateusz.
Thank you very much for this discussion.
Yeah, thank you. It's been very interesting.
Yes, and if you enjoyed the episode, please follow us on social media at neuroverse_pod and check out our website, neuroversepod.com.